All right, in our case study today, we're going to be talking about penicillin. Specifically, we're going to cover a few topics, including how penicillin can be used to kill bacteria. We're going to be putting a lot of focus on cell permeability in this case study, along with how the bacteria has now evolved to prevent the killing of the bacteria, so the resistance that it has over time to penicillin, with also a focus in cell permeability. And then lastly, we're going to talk about the bound structure of penicillin by looking at chimera and the enzymatic inhibition mechanism of penicillin. All right, so what's going to happen is penicillin works to burst the bacteria cell wall. And when it bursts that bacteria cell wall, water goes rushing in and the bacteria cell just bursts. So how does this exactly happen? It's directly acting against peptidoglycans. So those play an essential role in the bacterial cell structure. It's what keeps the cell wall and the structure of the cell on the outside really strong and keeps it not less permeable to any outside substances coming into the cell, like such as water bursting the cell. So they create sort of a mesh-like structure and the around the plasma membrane surrounding that cell, and it increases the strength of the cell wall and prevents those external fluids from coming in and entering the cell. So here's the structure of peptidoglycan, sort of in a more of a biochemistry aspect of that. And then we have here, no matter if the bacteria is gram-positive or gram-negative, it's still going to have that outside layer that it's providing. So whether it's providing the complete outside layer, and that's the outer shell, or whether it's a part of the cell wall sort of sandwiched in between the outer membrane and the plasma membrane. So again, we'll talk a little bit more detail about how penicillin is really bursting the bacteria's cell wall. When the bacteria multiplies, there's these small holes that open up inside of the cell walls, and peptidoglycans really fill those holes so they can reconstruct the wall. So what penicillin is doing is it's blocking the protein struts that really link the peptidoglycans together. So without that happening, the bacterium isn't able to close in those cell walls. So the peptidoglycan is really what sort of plugs those small holes, and penicillin is really binding to the it's really blocking that protein that's keeping the peptidoglycan together, so it can't plug up those holes. And then when that happens, the cell wall is much less permeable, right? So that's how it's really hurting that cell's permeability ability. So then when there's water concentration sort of surrounding that cell, and there's a high concentration compared to what's inside, then you're going to have that rush of water going into the cell through those holes that aren't blocked anymore, and then that bacteria is just going to burst. Alrighty, so now we'll talk a little bit about bacteria's resistance to penicillin. So bacteria is highly adaptable. It's highly adaptable because it can regenerate very rapidly and quickly, make genetic changes, and it can develop immunity in a few different ways. So it can develop immunity by having penicillinase. So that is bacteria producing penicillinase, which is an enzyme that degrades penicillin. So this ability can spread throughout the bacterial population using a process called conjugation, where individual organisms are sharing genetic information in between them. So we're sort of adding that genetic variability to the bacteria and giving us this ability to create penicillinase. We're going to be focusing a lot more so on the altered bacterial structure, so we'll get to that in just a second. And then penicillin removal, which is where other bacteria are developing systems to export penicillin. So bacteria has these pumps where they can release substances from the cell, so targeting releasing penicillin out of the cell. So when we're putting a focus on cell permeability, we want to talk about this altered bacterial structure. This is sort of what's giving the cell more permeability. So some bacteria can subtly change the format of their penicillin binding proteins in their peptidoglycan wall so that the penicillin can no longer bind to it. So what happens there? So what's happening is penicillin, so just to kind of go over what penicillin is doing and then sort of how the bacteria is developing the immunity, the penicillin is blocking transpeptidation. So penicillin binds to the penicillin binding protein, right? That's what we talked about, that receptor on the surface of the bacterial cell wall. So PBP is the receptor for substrate peptidoglycan precursor in bacteria. So antibiotics and penicillin can act as an alternative substrate that binds to that receptor and inhibits transpeptidase, which results in inhibition of cell wall synthesis. Right, so that was sort of what we talked about. The peptidoglycan can no longer plug up those holes because we're binding to that protein that really binds the peptidoglycan together. So how is bacteria developing this immunity? So it can alternate that receptor, that PPB receptor. So there's certain bacteria that might be resistant because they produce this beta-lactamase, but either due to altered PBP receptor or due to impermeability of their cell membrane to antibiotics, they can just alter that receptor. So penicillin is no longer able to bind to that, or it might generate over time where there's just an absence of the cell wall completely. So just lacking a cell wall, so that are really resistant to penicillin, right? Because penicillin is sort of attacking the cell wall when it's taking apart that bacteria. So without that, we don't have anything for penicillin to really bind to. So we'll look a little bit about the structure of penicillin as well. Let me open up my chimera here so we can kind of take a look at sort of this 3D structure. We can zoom in a little bit on it here so we can see some of these structures from within. All right. So lastly, we're going to just focus on the enzymatic inhibition mechanism of penicillin. So there are cross-linking peptide chains that provide the rigidity that to the cell wall, right? That makes it impermeable, or have a low permeability. The breaking of these peptide cross-linking is performed by an enzyme called transpeptidase. So the transpeptidase is helping with reforming the peptide bonds once restructuring the cell wall is done. Right, so we talked a lot about transpeptidase. So that's sort of how it is reforming and restructuring that cell wall. And sort of the mechanism of action that we're seeing from penicillin that's really attacking that is the penicillin acts by inhibiting transpeptidase. So it's inhibiting that enzyme. So by inhibiting that, the penicillin prevents the reformation of the peptide bonds. Thus makes the cell wall less strong, right? Without it being strong, then the integrity is lost, causing the bacteria to really leak out its cellular contents and perish. So it can leak out its cellular contents because the cell wall becomes very fluid, or what you'll see is water rushing into the cell of the bacteria and it bursting. And that is the end of my presentation. You can take a look at some of the citations here for more information.